Hello, thank you for joining us today on Science Spotlight. I will be speaking with Dr. Amy MacNeill at Colorado State University. Dr. MacNeill, why don't you go ahead and start by just telling us a little bit about yourself and the work that you usually do at CSU.
Hi, thanks for having me. Yeah, I actually usually do work to try to fight cancer with the immune system. So I use virus vectors similar to the ones that I'm going to be using for this COVID vaccine to try to deliver products into tumors that will stimulate the immune system to eliminate the tumor from the body. So I usually do cancer therapy, actually. My background is in viral pathogenesis and immunology. And I did a lot of work in a lab that did vaccine development at University of Florida many years ago. And so when the coronavirus pandemic began, I started thinking about ways in which I could use the same vectors that I'm using now that are safe in cancer patients to deliver a part of the coronavirus protein that would be safe as a vaccine against COVID-19.
So there are quite a few researchers that are working on different approaches to vaccines for COVID. So what is unique about your approach?
Yeah, I think it's really important that we have multiple different ways to approach this because we don't know what the best response will be until we try it. So my idea is to use a different virus that's attenuated that we know is safe and have it make just a small part of the spike protein of COVID-2, SARS-CoV-2. SARS-CoV-2, we know from research that we've done on the SARS virus that was initially a problem back in the early 2000s, and the MERS virus that was a problem after that. We know a lot from the research that we've done on those two very similar viruses. So we know that the spike protein that these coronaviruses have is needed to get into cells and allow for virus replication and allow for infection. So the idea is if we can block that protein from binding cells, then we can prevent infection completely. So the hope is by using a very small piece of that protein, we'll be making antibodies in the person who's vaccinated that are really good at just neutralizing that virus and stopping it from getting into cells, and not make any antibodies that might make the disease worse, because there is some concern about that with coronaviruses. They're kind of tricky little things. So there's all different ways to work on vaccines, but using that tiny bit of a protein is the way that I've decided to try.
So there's been a lot of information floating around that seems to be competing about whether or not people that are naturally developing antibodies against the virus are effectively immune from it. Do you know anything about that, and how does that relate to your work with spike protein specifically?
Yeah, there is concern, mostly from what we know about other coronaviruses, but we just don't know about SARS-CoV-2 yet. But from what we know about other coronaviruses and other species, there are some coronaviruses that you can make a vaccine against and it does great and it stops the disease. We've seen that in the pig industry. They had a horrible coronavirus outbreak several years ago, but we were able to stop it with a vaccine that was developed pretty quickly. But then there's coronaviruses like we see in the cat that we are worried that a really robust immune response actually makes it harder to eliminate the virus. So it allows the virus to get into cells that it normally wouldn't get into. And so we don't want to do that. We don't know if this corona is going to do that or not. We don't believe that SARS or MERS does. So we have high hopes that vaccines will be very effective if we target them to the right part of the virus. We hope and have some evidence that antibodies that are being made that people are infected and have COVID-19 seem to help or may help people that are currently sick with it. So transferring plasma to give antibodies to sick COVID-19 patients has had some benefit. So that gives us a lot of hope that vaccines will also have benefit.
So can you tell us just a little bit more about the cancer research that you usually do, and what ideas you were able to translate to this project?
Yeah, so I work in a field called oncolytic virotherapy. And so what that is using safe viruses that will replicate in tumor cells, but won't replicate in healthy cells of the body, to deliver immunostimulation or drugs or something that will help the body recognize the cancer as bad. Because one of the bad things about cancer is it's part of your own body that's grown out of control. And so it usually often, it can be hindered from fighting against it. So the idea is to allow these viruses in as replicating organisms that the body says, hey, that's not right, and it will help lyse the cancer cells because of that. And it's kind of it sounds like a way out there kind of way to fight cancer, but we actually do have some FDA approved oncolytic viruses that are out there for melanoma right now in the United States. And we have some in other countries as well. So triggering the immune response against cancer is a really exciting new avenue for cancer therapy that we've only just begun to really realize how important it is and what we can do to help patients with cancer this way.
And so has the cancer research that you were doing completely halted to focus on?
Good question. It is on a little bit of a hiatus right now. I have a small lab. And so we're really trying to focus on using these cancer vector viruses to start producing the SARS-CoV-S protein fragment. But we're always working on them. We're always thinking about them. So we're using this time to collate a bunch of the data that we had before COVID started and get it out there for the scientific community, get papers written, get grants written. So it's not as active as it normally is, but it's still moving forward.
So I noticed it looks like you're at home right now.
Yeah.
Home in the lab, how many of your lab personnel are still on board?
Yeah, normally it's only actually my research associate and myself right now. So most days I am in the lab all day. This is an exception for some personal things, but I'm also waiting on the next little set of primers that we need to come in the mail. So if they come in later today, that's where I'm heading, is the lab.
So speaking of waiting on things, what are the biggest challenges you've encountered in this project so far, and what challenges do you foresee or resources will you need that you don't have now to make this project successful?
Yeah, I've been really very lucky. My lab is set up to do this kind of vector production. So it's been a really easy switch over. So a lot of the things that I already had in place, I was able to move and use to fight SARS-CoV-2. So it's a fairly easy transition and a very quick one. I've gotten a lot of support from CSU to get some of these projects started and off the ground. So we're going to be able to get to a place where if it works, if it looks like it's promising, we can start getting larger pharmaceutical companies in place, hopefully by August, actually, and kind of be able, my hope is to be able to hand them over and give them to somebody who can bulk produce and make them available much more quickly than I can myself.
That's a really exciting timeline. I think we're probably all happy to hear that news
Yeah. There's
situation.
still a lot of steps after that, but it's been amazing how quickly we've been able to get these vaccines rolling out across the whole community. The fact that there are some groups in phase 3 clinical trials is pretty phenomenal right now.
So it's my understanding that there are quite a few COVID-related projects going on at CSU. Are you aware of some of the work that your colleagues are doing?
Yeah, there's a lot of exciting work going on. People are working on every aspect of the biology of this virus, every aspect of the epidemiology of how it spreads, as well as things like vaccines and maybe even medical treatments, drugs that could be given to stop replication of the virus once it's in your body. So real treatments for people who have it already. Vaccines are great for people who don't have it yet. But for people who have it, having those options to inhibit the virus spread is one of the things that groups are working on here. So we actually have a group through my department, the microimmunopathology department, that is involved in infectious disease and diagnostics and research. And they've really geared up and started working on this particular virus. That's their job. They're very good at doing that. They did it for Zika, they did it for MERS, and now they're doing it for this SARS-CoV-2. We have meetings and get together and talk about what each other has found out about the virus pretty frequently. And it's really exciting to see what everybody's doing. And the whole scientific community has been doing that for this pandemic. It's really been really collaborative and exciting to be part of.
Aside from pharmaceutical companies that you would need to partner with in order to scale up and conduct clinical trials, are there any other collaborations that you're looking for that would be helpful in your research?
Yeah, many of them are right here at CSU. A lot of the people who are doing the work to see what other species this virus can infect will be part of the team to test the vaccine and see if it's working or not before it ever goes into humans. So things like hamsters, for instance, are susceptible. So those groups are a very important part of this project. Also, the other groups that are doing vaccine research, I'm very interested in working closely with them as we begin to get to the testing stages to see if we can create antibodies. It may be that my virus is great to give the initial boost to the immune response, and then somebody else's vaccine will be better at then stimulating it so that it can be a long-lasting immunity. So using the vaccines in tandem is something that we're looking at to see what we can do, what we can get the best and most long-lasting immunity against this virus.
So CSU has a really prominent veterinary school, and you are also a veterinary pathologist. Are there unique opportunities for like doing animal testing ahead of clinical trials or anything like that at CSU that you're taking advantage of?
Yeah, there definitely are. You know, one of the things I kind of joke about, but I'm really not joking. I wouldn't mind if my vaccine isn't used in people ever, but it helps cats. So there are groups that are looking at how this disease affects domestic animals, our pets, our friends. And so some of the work we're doing is also aimed at protecting them.
Excellent. I won't take too much more of your time. Is there anything else that you want to share or messages you want to send out about the research that you're doing or that others at CSU are working on?
I just want to reiterate how important it was for us to have previous data from other coronaviruses to use to move this forward so quickly. So fund research, it's important. And I just want to make sure everybody knows how hard people are working to get this figured out. There's a lot of things we don't have answers for, but people are really trying hard to get those as quickly as possible.
Awesome. Thank you so much for sharing with us today, Amy. Thank you to all of you for tuning in. You can join our YouTube channel and subscribe for more future on the ground COVID-19 research updates.